Welcome viewers. Today in this session we will talk about the haploid genome organization in eukaryotes. The eukaryotes are those organisms in whose cell there are structures which are membrane bound. These structures are known as organelles. The term genome was first coined by the German botanist Hans Wencker in the year 1920. It means the total amount of genetic material present in a virus, in a bacterial cell, in an eukaryotic organelle, or the haploid set of chromosomes present in haploid organism. In previous days, the term genome was more precisely confined to haploid cells. But nowadays, the term genome has been redefined. In modern biology, the term genome denotes the entire genetic complement of a living organism, or it is the entirety of the organism's hereditary information. The branch of science which is dealing with the study of genome is called genomics. It studies the content, function, structure, organization of genetic information confined in whole genome. In this diagram, as we can see, the nucleus is an organelle which contains the genome in case of eukaryotes. Here, the DNA it is not present in the form of DNA double helix, rather it is packaged in the form of chromosomes which are rod-shaped structures and are confined in the nucleus of eukaryotic cell. Now let us move to the eukaryotic organism once more. The eukaryotic organism contains two types of genome. First is the diploid genome and the haploid genome. We know that the eukaryotic organisms are more complex in nature. As a result, they contain two different types of cell, the somatic cells and the gametes. The somatic cells contains two sets of chromosomes in its nucleus, and hence the genome present in somatic cells of eukaryotic organisms are of diploid type. Whereas the gametes of eukaryotic organisms contains chromosomes of only a single set, and hence the genome becomes haploid. So accordingly, we can say there are two types of genome. One is the haploid genome and the diploid genome, abbreviated by the symbol N and 2N respectively. As shown in this diagram, the haploid genome here is containing 3 chromosomes labeled or shown as pink, blue and yellow, and which are present in a single set. Hence the genome is becoming haploid genome. Whereas in case of diploid genome, the 3 chromosomes have been shown in pairs. These pairs are known as its homologous pair, and hence the genome becomes diploid as it contains 2 sets of chromosomes. So we can see how the genome can be broadly defined into haploid and the diploid type. Now let us move to the process which is occurring in eukaryotes, as a result of which the diploid genome is getting reduced to haploid. The division which takes place is known as reductional division or meiosis. So meiosis is the process in which the cells they reduce their diploid genomes to haploid genome. Now we will later on see why the importance of haploidy or why the genome gets reduced. So through the process of meiosis, as shown in this diagram, we can see that there are 4 sets of chromosomes, 2 small, 2 large. When these 4 sets of chromosomes proceeds through the different stages of meiosis, they become finally reduced to 1 set of chromosomes, each containing 1 long and 1 short chromosomes present in the 4 gamete cells. So as a result of here 4 chromosomes is getting reduced to 2, so the process is reductional division, which is taking place through meiosis. Now let us move to importance of haploidy, why meiosis is taking place in all of us, that is the eukaryotic system. The importance of haploidy lies in the fact that due to the haploidy, after the normal fertilization of gametes, the normal diploid state is restored back. Otherwise, it will lead to the production of abnormal human beings because the normal state will not be restored. Apart from this, we can study the important characteristics of gene and its effect on phenotypic characters when they are present in haploid conditions. Further on, we can study the role of mutation on genes and its effect on phenotypic characters of the mutant organisms. So in this way, we can directly study the gene and its effect on the phenotypic characteristics of a haploid organism. The term C means constant and it denotes the amount of DNA which is present in a haploid nucleus or half the amount of DNA present in diploid somatic cell of a eukaryotic organism. The amount of DNA is expressed in picograms. Later on in this program, we will see how the C value is directly or how it is related to the morphological or structural complexity of the eukaryotic organism. Now let us move to the genome organization in eukaryotes or how the DNA is packaged in case of eukaryotes. Eukaryotes, whether diploid or haploid, they are packaged or the DNA is packaged in them in the form of rod-shaped structures called chromosomes. The chromosomes are visible only during the cell division like during the mitosis or during meiosis. Now the next question comes in that why chromosomes are found only in eukaryotes, why not in prokaryotes? The content of DNA is far more greater in case of eukaryotes than in prokaryotes. In case of prokaryotes like E. coli, the length of DNA is of around 1200 micron long, while in case of eukaryotes, it ranges from 19,000 micron to 23,000 micron. And all this has to be fitted inside a nucleus which is only 5 to 10 micron in diameter. So as a result, it needs to be highly condensed. When it becomes condensed, then the length becomes shorter and the diameter of DNA becomes thicker. Hence, it becomes visible under the microscope. Now, the visibility of your chromosomes under the microscope is only visible, is seen during the cell division. Now, let us see what happens when the chromosomes proceeds towards the interphase. What is interphase? That is the stage between the two successive cell division. So, we are saying that chromosomes are visible only during the mitotic phase. As soon as the mitotic phase is finished, it enters the interphase. During this stage, the decondensation of chromosomes takes place.
Ma'am, how the supercoiled DNA accessed by the enzymes during DNA replication?
The supercoiled sense, we know that the chromosomes are highly coiled and hence they are highly supercoiled. So, most of the enzymes, they are inaccessible to this supercoiled DNA. But when the chromosomes enter the interphase, then what happens? Decondensation of chromosomes takes place. As a result of decondensation of chromosomes, the DNA becomes accessible to the enzymes and hence replication takes place during the interphase, which is also known as its S phase. So, S phase is one of the intermittent phase of interphase. And during this phase, the DNA, after getting decondensed from the chromosomes, is accessible by the replication enzymes. So, we said that the packaging of DNA into chromosome is more intricate and complex in case of eukaryotes. Now, let us see what are the levels of packaging. The DNA is packaged into chromosomes in case of eukaryotes in 3 distinct levels. DNA is packaged with the help of 2 main types of protein. First is the positively charged basic protein called histones, and secondly a large group of heterogeneous acidic proteins known as non-histone chromosomal proteins.
Ma'am, how are histones able to interact with DNA in chromatin to form nucleosomes?
Now what happens is chromatin, or rather the histones, are positively charged proteins. Now they contain in them positively charged amino acids like lysine and arginine to a greater extent, as a result of which they acquire a net positive charge. Due to this net positive charge, they are able to bind with the negative phosphodiester backbone of the DNA. As a result of this, ionic interaction takes place, and with the help of which the chromatin fiber is formed. We will now discuss about the first level of packaging, that is the formation of chromatin fiber. So what is a chromatin? The first level of formation is the chromatin. So when the chromatin is visible? During the interphase. Chromatin is visible under the light microscope as a beads on a string-like structure. Now, on closer examination of these beads, it shows that it is made up of a histone octamer. Histone proteins, as we know now, are the basic proteins. There are 5 main types of histones. One is H1, H2A, H2B, H3 and H4. The histone octamer is formed from 2 copies of H2A, 2 copies of H2B, 2 copies of H3 and 2 copies of H4. So there are 8 units, and thus it is known as an octamer. The DNA is wrapped on the surface of this octamer in the form of superhelical turns. So the DNA is wound around the octamer as 1.65 negative superhelical turns, and which in turn contains about 146 to 147 base pairs of DNA. So in this way, we can see how the histones are interacting with DNA to form a nucleosome. So what is a nucleosome? Histone octamer plus 146 base pairs of DNA wrapped around itself. So this is a nucleosome. The nucleosome beads, they measure about in 11 nanometer in diameter and 6.5 nanometer in height. And as a result, the DNA double helix from 2 nanometer diameter gets thicker to a fiber which is measuring about 11 nanometer in diameter. So we find the first level of condensation. The nucleosomes are connected to each other by means of linker DNA. The length of the linker DNA varies from 8 base pairs to 114 base pairs. It varies with the different type of species. The linker DNA is made, or it is accessible by the linker histone, which is histone H1 protein. The histone H1 protein helps in your gathering the nucleosome, or in for your going to the next hierarchy of the level of packaging. So in this way, we can see that the nucleosome is forming the repeating structure of chromatin. Now let us move to the different types of chromatin. The euchromatin, they are the more transcriptionally active regions of the nucleus. Now since they appear lighter in color, this means they are less condensed than they are in the mitotic chromosomes, and hence they are more accessible by the enzymes, which makes them transcriptionally active. On the other hand, the heterochromatin are more coiled regions and they appear darker. And since they are more coiled, they are less accessible by the enzymes, and hence they are transcriptionally inactive. Now let us move to the second level of packaging, or the hierarchy in the packaging of DNA into chromosomes in eukaryotes. The second level is the formation of 30 nanometer chromatin fiber, or it is also known as solenoid model. Now in this model, what happens? The nucleosomes are brought into close contact to each other to form a thicker fiber which is approximately 30 nanometer in diameter.
Ma'am, when the supercoils come closer to each other, don't the negatively charged phosphates repel?
Well, we know that 30 nanometer solenoid fiber model, the nucleosomes, they come closer to each other. However, there is always a chance of repulsion since the negative phosphodiester bond are coming closer to each other. But this repulsion is minimized because of the presence of histone proteins, as we know that histones are positively charged proteins. Apart from this, there are other proteins which also binds to the linker DNA and helps in this association or in close juxtaposition of the core nucleosome particles, as a result of which the next level of condensation takes place. In this next level of condensation, what is happening? So, the linker DNA gently curves as it connects the core nucleosome particles into a continuous helical array. So, there were two models hypothesized. One was the zigzag model, another is the solenoid model. But experimental researchers, they showed that after performing certain experiments, they confirmed that it is the solenoid model and not the zigzag model, which is the way in which the nucleosomes are held closer to each other in the 30 nanometer fiber. Now here what happens? The core or the linker DNA connects the core nucleosome particles into a continuous helical array, and each array or each turn is containing from about 6 to 8 nucleosomes. As a result, what is happening? The length of the DNA is getting shortened and again the diameter is increasing. So, the first level we saw the diameter increased up to 11 nanometer. In the second level, we are seeing it is increased up to 30 nanometer in diameter. So, hence it is becoming more thicker. The third level of packaging is the formation of chromosomes as we see them under the metaphase stage. In this, what is happening? Now, there are proteins in the nucleus which is known as the nuclear matrix protein, and one common example is the scaffold protein. We know that chromatin is held on to the scaffold protein. The shape of the metaphase chromosomes is determined by the scaffold proteins which forms the network, and here the DNA which is present in the 30 nanometer solenoid fiber contains sequences which are known as matrix attachment regions known as MARS, which bind with the scaffold protein. As a result, the solenoid model is folded into radial loops or series on the protein scaffold, and hence as a result the length is again shortened and the diameter increases to 300 nanometer. With further refolding and compaction of both the radial loops and the protein scaffold, the length of the DNA or length of the chromosome as an average is greatly shortened and the diameter increases up to 700 nanometer. This is the diameter of one of the chromatids of a chromosome seen during metaphase stage. So, let us once more enumerate the different steps in the packing of DNA of eukaryotes into chromosome. So, as we can see in this diagram, the first stage is the simple DNA double helix containing of a diameter of 2 nanometer. Now, this is folded in the form of, we know, the nucleosome, which is the first level of packaging. So, here the histone is getting packed with the DNA. The second level is the formation of 30 nanometer solenoid fiber model or the 30 nanometer chromatin. Now, this 30 nanometer chromatin is further bent into radial loops or domains which are held to the scaffold protein. This again is refolded to form a diameter of around 700 nanometer loops. Now, this 700 nanometer is one of the chromatid diameter of a chromosome. So, together it becomes 1400 nanometer in diameter and the length becomes greatly reduced, as a result of which the chromosomes are formed and seen or visible or made visible under the light microscope during the cell divisional stage, particularly during the metaphase of mitosis or meiosis. So, in this way we can see how the DNA is packaged with the help of proteins in case of eukaryotes. Now let us move to the structure of a eukaryotic chromosome. So we saw the eukaryotic chromosome. Let us now know about its structure. The structure of a eukaryotic chromosome varies with the type of species. It is generally rod-shaped, but according to the position of centromere, it can be divided. But let us now move to the substructures present within the eukaryotic chromosome. The first is the origin of replication. The second is centromeres, and the third is telomeres. So what are origin of replication? Origin of replication are the points at which initiation of replication in case of eukaryotes takes place. Centromeres, these contains kinetochores. As a result, kinetochores are proteins which helps in binding of the chromosomes to the spindle apparatus during the cell division. As a result of binding to the spindle apparatus, there is an equal distribution or segregation of genetic material between the cells, and hence the genome is, or the type of genome is made constant. If it is diploid, the cells after division remains diploid until and unless there is a reductional division. Then we have a telomere. The telomeres are the protective caps of the chromosomes and are present at the ends. Now they have got several fold functions. The first function is they prevent the exonuclease attack. The second function is they improve the stability of chromosomes. How do they improve the stability? By preventing the end fusion of chromosomes with the other chromosomal ends. They also prevent chromosomal aberrations from happening. They play an important regulatory role in cancer. So in this way, the telomeres, they form important function or serve several important function in the cell. Apart from these functions listed, there is another important function, that is they help to prevent, or rather they prevent the shortening of chromosomes after each round of replication. So the C value which is collected from the various datas shows that the C value in case of higher eukaryotes varies widely between species. There may be variation or there may be no variation among the closely related species of higher eukaryotes.
Ma'am, what is the significance of C value?
The C value, as the term suggests, it measures the total amount of DNA. Now with the improvement in technologies, now the C value details, they are being studied. With the help of C value data, we will be able to compare the species in a more phylogenetically manner, or how the genes they evolved and they were lost during the gradual evolution of a species. So in this way, the total amount of DNA is also correlated with the total number of genes, or with the amount of other DNA which plays an important structural or regulatory role or a functional role in the whole genome. So that's why the data of C value is very important in studying the genomic complexity of an organism, because the genomic complexity also represents the structural or morphological complexity of the organism. It has been found that in the eukaryotes, or rather in higher eukaryotes, the amount of DNA is not directly proportional to the structural or morphological complexity of eukaryotic organism. This is known as C value paradox. Then what happens to this huge amount of DNA? The huge amount of DNA does not always encode or code for proteins. Rather, they play an important structural or regulatory role in case of eukaryotes. These are known as repetitive sequences. The repetitive sequences were discovered with the help of DNA renaturation kinetics study. It showed that in case of eukaryotes, there are sequences which are repeated more than once in the genome, and hence they are known as repetitive sequences. whereas those sequences which are repeated or whose copy is only one in the whole genome are known as non-repetitive sequences. The repetitive sequences can be divided into moderately repetitive sequence and highly repetitive sequence. The moderately repetitive sequence, the copy number of such sequences are from 10 to 1000 times in the genome, whereas highly repetitive sequences are repeated more than 1000 times in the whole genome. The examples of repetitive sequences can be the pseudogenes or the spacer genes, or from the tandemly repetitive sequences to interspersed repetitive sequences. So these are all repetitive sequences not related directly to the gene function. Whereas the DNA which is related to gene function includes the ribosomal RNA, tRNA, or the histones we have, or on the one hand then we have the introns and the untranslated regions. Apart from this, the transcriptional regulatory sequences. So we can see how the repetitive sequences are coding for functional proteins, or how they are playing a regulatory role in the form or in the term of untranslated regions, in the form of transcription regulatory sequences, so that there is a control throughout the whole genome. Now let us move to the eukaryotic organisms whose genome have been sequenced so far. The first amongst them is Saccharomyces cerevisiae, the common yeast. Then we have Drosophila melanogaster, which is a fly. Then we have the nematode Caenorhabditis elegans, which is the nematode we know. A lot of work is being done on that organism. Then the Arabidopsis thaliana, followed by the common agricultural crop rice, scientifically known as Oryza sativa. Last but not least, Homo sapiens, that the human beings whose genome has also been completely sequenced. With the help of all the sequencing information, it has been proved that there are repetitive sequences, and these repetitive sequences, they work or they have a number of important regulatory roles, and they also satisfy the other information which we have gathered before with the help of other technologies.